Genomics has revolutionized our understanding of life. At the heart of this revolution lies sequence assembly. Imagine a giant jigsaw puzzle, but instead of a picture, it represents an organism's entire DNA sequence. Sequence assembly is the process of putting this puzzle together. This process is crucial for creating a complete picture of an organism's genetic makeup. It allows scientists to identify genes, study their functions, and understand how genetic variations contribute to disease. Without sequence assembly, our understanding of genomics would be incomplete. The importance of sequence assembly extends beyond basic research. In medicine, it aids in diagnosing genetic disorders, developing personalized therapies, and tracking the spread of infectious diseases. In agriculture, it helps improve crop yields and develop disease-resistant varieties. The applications of sequence assembly are vast and continuously expanding. There are two primary approaches to sequence assembly: de novo assembly and reference-based assembly. De novo assembly is like solving a puzzle without a picture. It is used when the organism's genome has not been sequenced before. Scientists rely on overlapping sequences and complex algorithms to piece together the DNA fragments. Reference-based assembly, on the other hand, is like solving a puzzle with a picture as a guide. It involves aligning the DNA fragments to a closely related organism's known genome sequence. This approach is faster and computationally less demanding than de novo assembly. The choice between de novo and reference-based assembly depends on the availability of a reference genome and the research goals. De novo assembly is more challenging but provides a complete and unbiased view of the genome. Reference-based assembly is faster and more efficient but may miss unique or highly divergent regions. Sequence assembly, while essential, is not without its challenges. One major obstacle is the presence of repetitive DNA sequences. Imagine trying to assemble a puzzle with many identical pieces. It becomes difficult to determine their correct order. Similarly, repetitive DNA sequences can confuse assembly algorithms, leading to errors in the reconstructed genome. Another challenge is sequencing errors. Sequencing technologies, while advanced, are not perfect. They can introduce errors like misreading a DNA base, which can propagate through the assembly process. These errors can lead to misassemblies and inaccurate gene predictions. Furthermore, low sequencing coverage, meaning not all parts of the genome are sequenced equally, can create gaps in the assembled genome. These gaps are like missing pieces in a puzzle, making it difficult to get a complete picture. Addressing these challenges is crucial for obtaining accurate and reliable genome assemblies. Section 4. Overcoming repetitive regions and sequencing errors. Scientists have developed strategies to overcome the obstacles posed by repetitive regions and sequencing errors. Long-read sequencing technologies have emerged as a powerful tool for resolving repetitive DNA. These technologies generate longer DNA reads, spanning repetitive regions and simplifying their assembly. Specialized assembly tools have also been developed to handle repetitive sequences. These tools use algorithms that can recognize and resolve repetitive regions based on their unique characteristics. By combining long-read sequencing with specialized assembly tools, scientists can significantly improve the accuracy of genome assemblies. To combat sequencing errors, error correction algorithms have become indispensable. These algorithms identify and correct errors in the raw sequencing data before assembly. By minimizing errors at the outset, error correction algorithms enhance the reliability of downstream analyses. Section 5. Addressing low coverage, heterozygosity and chimeric reads. Addressing low sequencing coverage, heterozygosity and chimeric reads requires additional strategies. Increasing sequencing depth, meaning sequencing the same DNA region multiple times, can effectively address low coverage. By increasing the number of reads, the likelihood of capturing all parts of the genome improves. Heterozygosity, the presence of genetic variations within an individual, can also pose challenges. Specialized software tools have been developed to handle diploid and polyploid genomes, accounting for genetic variations and improving assembly accuracy. Chimeric reads, which are formed by the artificial joining of DNA fragments from different parts of the genome, can lead to misassemblies. Filtering out chimeric reads during pre-processing and using assembly algorithms that can identify and resolve such anomalies are essential for obtaining accurate assemblies. Section 6. Tackling short read lengths and computational demands. Short read lengths and computational challenges are inherent to sequence assembly. While long read technologies are improving, short read sequencing remains widely used. To overcome the limitations of short reads, scientists use scaffolding techniques. Scaffolding involves linking together assembled DNA segments called contigs, using additional information about their relative positions. Computational challenges arise from the sheer volume of data generated by sequencing technologies. Sequence assembly is a computationally intensive process requiring significant memory and processing power. To address this, researchers utilize parallel computing, distributing the workload across multiple processors to speed up computations. Cloud computing has also emerged as a valuable resource for genomics research. Cloud platforms provide access to vast computational resources, allowing researchers to perform large-scale sequence assemblies efficiently. As sequencing technologies continue to advance, generating even more data, computational efficiency will remain a key focus in sequence assembly.